Morning. Well, the power of one. So what's the power of a single genome? And here's the challenge. I have 10 minutes to talk about a single genome that is 10 to the 11th base pairs. So I think making it simple might be a little difficult, but we'll do it. We'll do our best, and here goes, one off the hip. So I'd like to focus on cancer and personalized medicine, and a definition of cancer is that it's a genetic disease driven by aberrant genetic programming. It's highly heterogeneous in any single individual. It's rare that there's a single clonal tumor. It's characterized by changes in genomic sequence. And a couple of important things here is that there are germline mutations, such as BRCA1 and 2, where you inherit the propensity to have cancer. And the more common, somatic mutations, where up to 90% of cancer genes fall into that category. By 2011, it's important to note that there have been 436 oncogenes or causative mutations identified. So what's personal medicine in light of that? Well, personalized medicine is a healthcare strategy that optimizes cancer care and outcomes for each individual patient, and it's based on the genomics of the disease. In other words, what's the tumor genome telling you about the way that it might be addicted to certain pathways of survival? And if you can interrupt those, well, you could probably treat very effectively a very small number of people. So personalized medicine, the way to look at it, if you have a population of patients with cancer, they're highly heterogeneous, but you may find similar mutations in subpopulations. And I think the first challenge that we face in pharma is how do you develop, create drugs for subpopulations and be able to come in with a price that is achievable from a reimbursement standpoint, and therefore be able to keep investing in this particular genetic chase. The goal of personalized medicine is that it's targeted to specific oncodrivers. You identify patients through molecular testing. Oops, there's something new. You got to be able to find the patient. You can't just seed the product and hope that some patients benefit. Like in the old chemotherapy era, which is still very prevalent and important, if you treat 100 patients, you may get 20 who respond. The hope with personalized medicine is if you treat 100 patients, 80 respond. And there's the value proposition. But it also impacts genomics-based drug development, and here you have a similar challenge where you treated in the past, like I say, 100 people and very few respond. The whole idea here is to be able to identify and stratify patients on that genetic fingerprint of their tumor and develop drugs for those specific genetic mutations and pathways, and thereby produce a highly segmented or molecularly segmented approach to disease. So is that the new paradigm or the bus to catch, is molecular segmentation? With personalized medicine, that's indeed what is implied. However, before I get into some of the early successes, there's been a recent New England Journal of Medicine publication that says that when you look at a tumor and then you look at its metastases, you're not finding one genetic fingerprint, you're finding many. So now there's an n equals 1, there's a 10 to the 11th, and then within the 10 to the 11th, there are even sub-pockets of different things, different cells acting in different ways. So how many personalized medicines will you actually need to succeed? And so there are more challenges yet with that. However, if we go back to where we stand today and maybe the challenge of that New England paper that's been recently published, you start with a driver mutation and a discovery, you develop a proof of concept, and then ultimately you hope to approve your drug. If we look at BCR-ABL, its discovery, the Philadelphia chromosome, about 40 some odd years ago, and the development of Gleevec to treat that disease to approval. Well, it's 41 years from understanding some genetic or molecular aspect of the disease to bringing a drug to market. If we look at PARP inhibitors, we're looking at 15 years. If we're looking at ERB-B2, we're looking at 13 years through that same process. If we look at HER2 as an example, 19 years from discovery of the HER2 mutation to an effective treatment, 19 years. And with BRAF, it's 8 years. One company's been able to narrow that down. With Pfizer and ALK inhibition, it's 3 years. So the ability to identify the right mutation and the right pathway and deliver a treatment that works is being reduced. So to those who worry about the cost of R&D and how quickly can you get to beyond proof of concept, this is an extremely encouraging cost-saving approach to drug discovery and development. However, the druggable targets in solid tumors and response rates in biomarker-driven trials, there are very few of these drugs. But when you look at on the right side of it, gefitinib, crizotinib, imatinib, PLX 4032, the PARP inhibitors, Herceptin, and mitomycin C, not yet tested, response rates vary from high 40% up to 73%. So these are very significant gains in treatment efficacy. Now, some of the challenges that we're going to face, and I mentioned already earlier, how are you going to find these patients when you have a medical infrastructure, and in particular in Canada, one that is not actively testing for molecular markers and biomarkers for oncogenes. If you take a look at the dark blue columns in the histogram from 2000 to 2007, there's certainly more disease genes discovered, not only in cancer, but across all diseases that are molecularly driven. And the molecular tests that are available in North America almost match these. You know, somewhere in 2,400 or 2,300 genes and about 1,800 tests, most of these research tests. And if you take a look at that tiny little purple column, you see the actual number of molecular tests available in Ontario labs in 2007. This is changing, but as a commercial person or someone responsible for business development or bringing one of these medicines to the market successfully, you not only have the challenge of convincing the clinician to want to prescribe the medicine, you have an infrastructure challenge of being able to get patients screened and tested. Okay, so what's the rate of attrition of phase 1 trials of anti-cancer drugs to get to registration? So here we go. Okay, almost half the audience has a sense of how much failure there is, and that if you're working in this kind of area, if you're on the R&D side, you fail more often than you actually win, much like baseball, a baseball analogy. I don't like baseball, but it's a good analogy. Question 2, now if you look at TKI type drugs, which are targeted to specific pathways, and if you can actually identify a subpopulation, what do you think the failure rate or the rate of attrition before a drug gets to registration in this type of R&D framework? There we go. Ooh. And to registration. So there is hope and there is proof of concept here that personalized medicine could work. Now here's the marketer's challenge as well. So maybe you can segment your patient population